Hello everyone. Good evening. Welcome to this class. Welcome to this video. And today we'll be talking about a quick concept regarding CSIR-UGC NET examination, and this will help you to acquire the knowledge regarding mRNA transport mechanism. And mRNA transport mechanism is a very important topic for CSI examination, and this is sometimes avoided in some discussion, but this is very important, and you need to know how this transport mechanism works, how the transport works. Okay, so I will briefly explain it to you, so you need to remember everything, as it is a molecular biology topic. So this is all about molecule. So as you know, the mRNA, after its transcription, so it needs to be transported, or needs to be exported to cytoplasm. But mRNA in its RNA form, like only the mRNA, it cannot be exported to the cytoplasm. It is not exported, it is not being exported to cytoplasm. So what does it need? Okay, so mRNA needs some other components which will be attached to the mRNA, and then this whole complex, okay, this will be transported, or this will be exported to the cytoplasm. Okay, so the mRNA, when it will be exported, so we will name that form as mRNP, messenger ribonucleoprotein, because some proteins will be attached to the mRNA. And what are those proteins? Now let's discuss. So mRNA must be attached to one important protein, and you need to remember the name of the protein, that is MEX67 MTR2. This is showing in the picture. Okay, MEX67 MTR2. This is the protein which needs to be attached with the mRNA, and together it will help the mRNA to be exported to the cytoplasm. But not only MX67 MTR2 will be attached to the mRNA, some other proteins will be associated, because this MTR2 MEX67 or MEX67 MTR2, this protein, okay, this protein will be attached by the help of other protein. Now let's discuss step by step. So you need to see this. At first, two proteins, or rather we can say this is a complex. This is a complex named TREX. The full name is transcription export complex. So if you get the question regarding this topic, so they will not mention the full name, or they will not ask you about the full name. Okay, the TREX is transcription export complex. The transcription export complex consists of three proteins. One is sub2, another one is THO. So you don't need to remember the full name of these proteins, and YRA1. So this TREX will help the MEX67 MTR2 to be attached with the mRNA, and you can see THO, that is we have seen here THO. First, THO will attach sub2 to the mRNA along with YRA1. So sub2 and YRA1 is now attached. But when MEX67 and MTR2 needs to be attached, then sub2 will be removed, and as well as YRA1 will be removed too, and YRA1 then leaves the space for MEX67 MTR2 binding. Now MEX67 MTR2 will bind to the mRNA, and together it will form a complex like this, a mRNP like this. This is that mRNP which will be exported to the cytoplasm from nucleoplasm, or we can say from nucleus. From nucleus, this mRNP will be transported to cytoplasm, and the size of the mRNP will be 35 nanometer, very less. Then after forming this mRNP, or rather we can say after the formation of this mRNP, now this mRNP needs to be transported through a barrier, one channel, that is NPC, or nuclear pore complex. What is that? Nuclear pore complex. So nuclear pore complex. So I will suggest you to write these notes, okay, in your copies, so that you will remember, and you can write in your own way, okay. That I leave that up to you. So nuclear pore, it must be transported through a nuclear pore complex, and as you know, nuclear pore complex consists of several proteins, several NUPs, okay. So we will say nucleoporins. That nucleoporins or NUPs will help in this transport mechanism or export mechanism, okay. So there are two components, as you will see. The green components are the green components, this one. These green components are integral part of nuclear pore complex, and one component named MLP1. So MLP1 will help the binding of NAB2. Now NAB2 will bind. NAB2, this is an important protein. It will bind to the mRNP, which has formed earlier. So NAB2 will bind to the mRNP, and this binding NAB2 and mRNP binding will occur at the side of nucleoplasm. On the side of nucleoplasm, this mRNP NAB2 will bind. Now this whole complex, mRNP along with MEC67 MTR2 and NAB2, so it will pass through this nuclear pore complex, and very important point, MEC67 MTR2 will form some binding, or will form some attachment with FG residues of NUPs, nucleoporins. FG means phenylalanine and glycine. Phenylalanine glycine residue present in the NUPs, in the nucleoporins, and MEC67 MTR2 will form an attachment or form a bonding with FG residues, okay. And this is important. This is a very important point, and this can be asked. So after this, then the mRNP now reaches this mRNP reaches on the side of the cytoplasm, okay. So now this complex reaches to the cytoplasm, and there one important protein, and this is an very important, this is an DBP5, this is an ATPase, or you can say this is an helicase, or rather you can say ATPase, okay, no, ATPase. So this DBP5, that is the ATPase. So this ATPase now, with the help of IP6 and GLE1, and these proteins usually present in the attachment with NUPs, specifically NUP 159. So NUP 159 and DBP5 and IP6 GLE1, these proteins are present there in association with each other. So this DBP5 now with the help of its ATPase activity, okay. So now it removes, this DBP5 removes NAB2 and also MEC67 MTR2, and now the mRNA will be free of these proteins. So now there will be no mRNP, now we will get free mRNA on the side of cytoplasm. On the side of cytoplasm, we will get free mRNA because DPP5 removed the NAB2 and MEC67 MTR2. So in this way, the mRNA will be transported with the help of some very important proteins, and the name of the proteins which I mentioned in this video, these are most important proteins, and usually question can be asked from these proteins only. Question can be asked with regard to these proteins only, not any other protein, because these are the main proteins. These are the primary proteins required for the mRNA transport. Okay, I hope you understood. And another theory or another finding, they says, the group says that this MEC67 MTR2, they are being attached with the mRNA inside the NPC. But like whatever the thing, MEC67 MTR2 must be attached with the mRNA, otherwise this transport mechanism can't happen. And also MEC67 MTR2 and FG residue binding is very important for this transport mechanism, because MEC67 MTR2 containing mRNP, usually it gets passed through the NPC pore, or NPC, we can say NPC channel, because it's a channel, NPC channel. Nuclear pore complex forms a channel. Forms a tube through which this mRNP will be transported from nucleoplasm to cytoplasm. Okay, so this is the concept. I hope you understood. If any question arises regarding any point or any protein, you can ask me in the comment section. Okay, so just keep learning, and along with this you need to solve questions too. Okay, if any doubt appears in your mind, then ask me right away, and ask me your doubt in the comment section, in the Telegram channel. The links are there in the description, so go there and ask your questions, send your questions to me. So I'll be happy to answer your question. And for quick purposes, you need to ask the questions in the comment section. Thank you so much. So let's meet in the next video, and till then, bye, take care, see you in the next video.